This year, I decided to focus my lectures on what are known as transposable elements or mobile elements. These are a large fraction of most genomes and have been variously termed selfish DNA, junk DNA, DNA that doesn't seem to serve a purpose. Many of these elements can no longer move. They're dead or inert transposable elements, but some of them can still propagate. They can still copy themselves and move around using various strategies. The mobile versions of them are extremely dangerous to a genome because these elements can duplicate themselves. They can move around either by copy and paste systems or through an RNA intermediate. And they can reintegrate into different parts of the genome and cause mutations, they can cause damage. So these elements, in some contexts, are thought to have even derived from viruses. And they're termed as being endogenous viruses. Some of them are endogenous retroviruses. They're there, present. Up to 50% of the human and mouse genome contains such repeat elements. They've been called parasites, but on the other hand, we now know that they can actually also be helpful. The reason I want to talk about this fraction of the genome that doesn't seem to serve a purpose at first sight is because it's very, very tightly linked to epigenetic processes. And epigenetics are the changes in gene expression or genome function that can occur without affecting the sequence of DNA. It seems that these elements can attract repressive epigenetic factors. In other words, the transposable elements or their relics seem to be able to bring in the machinery that will keep parts of the genome quiet. And so now what people are realizing is that maybe these elements actually have served to nucleate the more heterochromatic or silent parts of the genome, the parts of the genome that need to be kept quiet. Whether they need to be kept quiet because it's a gene that can only be expressed at a particular stage of development, or whether it's a part of the genome that should almost always be quiet. And another feature of these mobile elements is the realization that because they can probably attract epigenetic factors, they can be used to create epigenetic processes. For example, imprinting, genomic imprinting, whereby depending on which parent transmits a gamete, a gene will be on or off. So imprinted loci are only expressed from one chromosome, either the paternal or the maternal chromosome. And it's becoming recognized that maybe what creates this differential state in the germline is partly due to these repetitive elements that are somehow recognized only in the paternal or only in the maternal genome as being different. And it's this that somehow allows such loci to evolve. The other process that I'm very interested in and that my lab actually works on is X chromosome inactivation. And this is a process which is classically known as being epigenetic because one whole chromosome in females, one whole X chromosome is shut down transcriptionally, it's silenced. The DNA of that chromosome doesn't change, but the chromosome is no longer expressed, it no longer produces RNA and protein. And the person who discovered it, Mary Lyon, in the early 1960s, actually went on to propose that one of the key features of this process might be a role for line elements, repeat elements. So transposable elements that seem to have accumulated on the X chromosome. And what she proposed is that these elements might help the process of X inactivation. In the 1930s and 1940s, an American cytogeneticist, Barbara McClintock, discovered, just by looking down the microscope at what happens in maize, that many of the unusual phenotypes, changes she was seeing in the color of grain, for example, were due to what she proposed were mobile elements or transposable elements. She realized that there were certain changes that led to chromosomes breaking that she could follow genetically and cytogenetically by looking at the chromosomes down the microscope. And she realized that these changes also seemed to affect the expression of genes. And she was really visionary because she realized that this was DNA that could potentially move. In 1983, she received the Nobel Prize for her studies. But gradually it became to be realized that this wasn't just a phenomenon in maize. People started to realize that even in bacteria, there were such elements that could move around. And progressively in other organisms. And in particular, the sequencing of the genome from 2000 onwards made everyone realize that actually most genomes are full of these sorts of elements. These are all very new and exciting fields, but indeed it's been a sort of a revelation, I think, to the scientific community that this so-called junk DNA is in fact extremely important and useful, and it shows just how correct Barbara McClintock was when she proposed that these elements, these mobile elements, could indeed be controlling elements in our genome.